Today we have Professor Christopher Rudd at the University of Cambridge in the United Kingdom here today. Professor Rudd, it's nice to have you here today. Could you tell us a little bit about the development up to the T-cell paradigm?
Yes, well, thank you. Yeah, it was an exciting time during the 1980s when it became clear that there were certain receptors on T cells that could be ligated with the use of monoclonal antibodies that allowed for the study of how T cells become activated. That is, the signaling pathways that are used by T cells, T cell receptor and its co-receptors, to activate gene transcription, leading to cytokine production and the proliferation and the function of T cells. And my involvement started with an interest in tyrosine phosphorylation because that really hadn't been defined at all in T cells. And really all that was known was that there was a SARC family member called PB60 SARC that was associated with middle T antigen, and in the case of the Rous sarcoma virus, was activated to cause cancer. But no one had really defined a function for these family members in the control of normal cell growth. And so Larry Samuelson had identified within the T cell receptor complex a site that was phosphorylated on tyrosine residues. But again, no one had really worked out how this process was initiated. And so I reasoned that perhaps instead of the T cell receptor, which lacks these cytoplasmic tails needed to activate cells, instead of it interacting with a putative kinase, perhaps co-receptors such as CD4 and CD8 were involved in the activation process. And however, if you looked at these co-receptors, they also lacked homology with kinase domains, and so it wasn't obvious how they would be implicated in initiating a phosphorylation cascade. So, however, given that CD4 and CD8 bind to MHC antigens coordinately with the T cell receptor recognition of MHC, that is to say, CD4 binds to MHC class II antigens and CD8 binds to class I antigens, it seems a reasonable possibility that they may be the mediators of the activation needed for the activation through the T cell receptor. And it became readily evident when we began to look at these receptors that they were associated with a high level of kinase activity, which we then mapped to tyrosine, and this led to the phosphorylation in immunoprecipitates using anti-CD4 and CD8 of a band at about 56 kD that was tyrosine phosphorylated. And around that time, a kinase a couple of years earlier had been described by Jamie Marsh as a kinase called P56LCK. We managed to get an antibody to this and managed to show indeed for the first time that CD4 and CD8 were associated with P56LCK, and it was an active kinase. We then showed that it phosphorylated components of the T cell receptor complex, and that was really the first evidence, first of all, implicating tyrosine kinases in the activation of T cells and in activating a tyrosine phosphorylation cascade. And this cascade then led to the identification of another kinase called ZAP70, as well as a whole array of intracellular substrates that are tyrosine phosphorylated, which really are the mainstay of work on T cells, mediators of T cell function presently today. So it's widely accepted now, I think, that CD4 and CD8 binding to LCK are the initiators of the T cell activation cascade.
Thank you very much, Professor Rudd. This was a very interesting topic to cover.